Hello. In this video we'll be looking at some applications associated with the simple models of the particle in the box. We'll be looking at something called FEMO theory. I will define that in a second. And we'll be applying it to both the one-dimensional cases and the two-dimensional cases. And as you might imagine, there are also situations where the three-dimensional model could also be employed. So, FEMO, what does that represent? The word or the FEMO stands for free electron molecular orbital model. So what we're going to be doing is, as mentioned in class, is associating wave functions with orbitals and using the general rule for orbitals in order to determine certain properties of these systems. We're going to be applying the model to conjugated pi systems. We have pi electrons that are delocalized in a network. For linear chains, we're going to be looking at a situation where we're going to very simply apply this to the one-dimensional box. So, for example, we have something like hexatriene here, and we're just simply going to be setting our box length from 0 to A as corresponding to the length of the chain itself. And we're going to have pi electrons delocalized over this region from 0 to A. So those electrons are going to be treated as the particle in the box corresponding to our network. So we will have, of course, with our particle in the box, the energy levels associated with quantum number A and related to the mass, m being, of course, the mass of the electron in this equation, and A being the length of the chain, which is essentially the sum of the bond lengths, as we see from 0 to A. So we're going to determine our length by summing up the bond lengths here in this model. Alright, so in this model we're also going to be using to determine what's called the HOMO-LUMO transition, which is going to be, when we take a spectrum, it's going to be the longest wavelength, lowest energy transition between two levels in the particle in the box model. HOMO corresponds for the highest occupied molecular orbital, and LUMO corresponds to the lowest unoccupied molecular orbital. Alright, so if we have these three levels that we show right here, and let's say we have 4 electrons, we're going to fill in 2 electrons into each level, and the highest level being n equal 2 in this case would correspond to our HOMO, and then the lowest level, which would be n equal 3, which is unoccupied, is called our LUMO. So this transition between the two, as I mentioned, is going to be the longest wavelength, lowest energy transition associated with this system. And again, to a first approximation. As we know, the systems are always, the real molecular systems are more complicated than this. So we show here that transition from the n equal 2 to the n equal 3 level in this situation. And after the fact, we're going to end up with 1 electron in the n equal 2 and 1 electron in n equal 3, as we have here. That corresponds to an excitation and an excited state. So we're going to calculate the wavelength associated with this transition. We need to know the energy difference between the n equal 2 and the n equal 3 levels. And that's going to, of course, be that energy difference, going to be the energy of our photon, which, of course, is going to be related to hc over lambda, the normal equations that we use for these kinds of things. Alright, so let's look at a specific example. So here we have butadiene. Alright, and how many pi electrons does butadiene have? Well, there's 1 pi in our network. There's 1 pi electron for each carbon, or if you'd like, 2 electrons for each of the pi bonds that are shown here in the localized picture. And of course, you know from organic chemistry that everything is delocalized. These pi electron systems are delocalized. So we have a total of 4 pi electrons. in our system. So, again, applying the one-dimensional particle in the box model, we have 3 levels, n equal 1, n equal 2, n equal 3. And again, as we just did on the previous slide, since we have 4 electrons, we fill them into electrons so that our HOMO is, as was before, n equal 2, and our LUMO is n equal 3. All right. Now, there is going to be the transition. So we've got then the question of how long is our box? Well, the average bond length in these pi systems is about 140 picometers, and since we have 3 bonds in the butadiene molecule, the total length of our box is 420 picometers. All right. So then to calculate the energy difference associated with those, we just use the n equal 3 associated with the LUMO and n equal 2 associated with the HOMO. So we plug those numbers in, as we see here. We use the mass of an electron, and here's our length of the box, and we get an energy of 1.71 times 10 to the minus 18 joules. We calculate the wavelength associated with that. It comes out to 116 nanometers, and which is in the ultraviolet region of the spectrum. So the butadiene is not have any color associated with it. This would be the longest wavelength, lowest energy transition, at least according to this particular rather simplistic model. But it does predict that we won't have any visible absorption, which means there will be no color associated with this particular molecule. All right, well, let's look at a molecule with a longer conjugated chain and one that has color to it. And this is beta-carotene, as indicated here. We have this conjugated chain between the 2 rings at the end. And if we go through and we count the number of bonds, we find there are 21 bonds in that conjugated chain, and again in the localized model, 11 pi bonds. Of course, we know it's all delocalized, but that helps us to figure out how many electrons we're going to have in our system, and so we're going to go ahead and then take a look at that. So in this situation, so we have 11 pi bonds in this, and that means we've got 22 electrons in that chain. And that means we've got 22 carbon atoms in that chain. So again, each one contributes a pi electron. So we have 22 electrons, and that means filling up 2 electrons per orbital. We're going to have 11 orbitals that are filled, and so that means that our HOMO is going to be n equal 11. The LUMO is then the next one above it. It's going to be n equal 12. So we go through the process we did before. We figure out the length of the box. There were 21 bonds, if you remember, as we counted. So we multiply 140 by 21 to get the length of our box. And then we plug it into the equation. In this case, we're using the equation for the difference in energy between adjacent levels as being 2n plus 1. Let me just write that out here so it's very clear to you which equation we're using. It will give us the same as result as before. This was an equation we derived on a previous video. So again, the energy difference between 2 adjacent levels, between n to n plus 1, is going to be given by this equation. Again, it's something you should be familiar with. It's one that we've looked at before. And so again, we calculate an energy difference of 1.6 times 10 to the minus 19 joules, and calculating the wavelength is 1,230 nanometers. And so we have this situation where we are in the infrared. So the longest wavelength transition here is the infrared region of the spectrum. Again, this is an approximation. We know the beta-carotene has a color associated with it. It's the color, it's the pigment in carrot, so it has that orange color. The reality is this particular prediction gives us a wavelength that is longer than the actual wave. Sorry for the interruption there. Let me just repeat this. The 1230 nanometer is an infrared absorption, and in reality we know that beta carotene, the longest wavelength transition to beta carotene is actually in the visible region, so this is a little bit longer than the actual one. But the thing about the FEMO model is it does a very good job of predicting trends. While the numerical values may not be terribly accurate, it does predict trends very well. And one trend that is particularly important is this trend that we've noticed just in comparing butadiene to beta carotene. Butadiene, you'll recall, had a wavelength for its HOMO-LUMO transition as being 116 nanometers. Which is much shorter wavelength than this case of beta carotene. So one thing we're going to note is that as the length of the chain increases, the wavelength associated with the conjugated chain also, for the HOMO-LUMO transition, also increases. So as the length increases, the wavelength increases, which actually explains behavior that we see in reality. And certainly one of the things that you're going to be testing in the experiment associated with the dye molecules in the lab is noticing that as the length of the chain does increase, we're going to see the wavelength increasing. So for example, butadiene exhibits no color, but as we shift to longer wavelengths in these conjugated chains, and longer chains and longer wavelengths, we're going to see that these molecules, as they get longer, start to absorb visible light and actually then have some color to them. So again, as we increase the length of the conjugated chain, we're going to see a shift in the HOMO-LUMO wavelength to larger values, to longer wavelengths. This is a trend that the particle-in-a-box model, while not terribly accurate when it comes to wavelengths, because it is a very primitive model, does a good job of predicting with these trends. So again, this slide here just shows a relationship between beta carotene and vitamin A. If we take the beta carotene molecule and split it into two, we get the three different systems that we see here that just simply differ with the functional group on the end of that particular chain, and of course this is particularly important when it comes to the vision mechanism. This shows the molecule retinal, which you'll recognize again. You have the cyclic group on the end and that conjugated chain here. This is the cis form of the retinal, which is the form that is the original form that then couples with rhodopsin in the vision process. When a photon of light hits this molecule, it causes it to undergo a cis-trans isomerization reaction, which then, as a result of that structural change, leads to the sequence of events that registers as vision in the brain. And of course, as we mentioned earlier in class, that photon of light essentially breaks the pi bond and allows it to rotate to go to the trans conformation. If we look at the energy of the coordinate system, again we talked about this in class, we'll note that we have this particular energy where we see on the left side the little minimum there corresponding to the cis, and there the deeper minimum trans on the right side, and that's because the trans is a more stable conformer. And this process of rotation actually causes the molecule to surmount the activation barrier and allow it to proceed to the other side. All right. Now here is another particular example that we're going to be working on in class, and this is a situation where we're actually looking at a two-dimensional model, because we can consider pi electrons in these metal porphyrins to be like a two-dimensional box, and so we can take that and do some calculations. If we have 26 pi electrons in this system, in this two-dimensional box, where the side of the box, and we're going to assume it's square, is 1 nanometer, we can calculate the HOMO-LUMO transition using the equations for the two-dimensional model. So here is just a kind of a picture of this model that we're talking about, where we do have, so here is our box, essentially, right here. Okay, and you can see the pi electrons associated with the pi conjugation associated with, if you look at the pi bonds here. We have this conjugated chain that wraps around that particular box. So in this model, to determine the HOMO-LUMO situation, we need to figure out what the highest occupied molecular orbital is. It's not quite as straightforward as was the case for the one-dimensional box. In the one-dimensional box, if we had 26 electrons, we knew we would fill 13 orbitals, and then that meant the HOMO was n equal 13. In the two-dimensional box, we have 2 quantum numbers, and then we have to remember that we have the square box degeneracy. So then we have the 1, 2, and the 2, 1, and then we have the 2, 2, and we fill in electrons, and we just keep going, filling in electrons until we've filled in all 26 electrons. So what you have to do is you have to figure out the structure, filling in the electrons, 2 electrons per orbital, until you get filled in 26 electrons, and you figured out which level corresponds to the HOMO, and then the one right above that, of course, is going to be the LUMO. And so that's what we'll need to do in that particular system to figure that out. And again, we'll be working on this particular model in class. So stay tuned for that.